it's a series of steps. And those steps are classified into three types. First step is glycolysis, second is Krebs cycle, and third one is electron transport, which were discussed in our previous classes. And today we are going to discuss the summary of production of ATP during aerobic respiration. It means total calculation of energy currencies in the form of ATP molecules. Okay, so if we observe the first step of this aerobic respiration, that is glycolysis, that produces two ATP molecules and two NADH2 molecules. We know that during the during the glycolysis, in the starting of the glycolysis, we have seen the two ATP molecules are utilized and two more are produced for each one glyceraldehyde phosphate. And there triose phosphate molecules are two. For each one triose phosphate molecule, two ATP molecules are synthesized. So totally 4 ATP molecules are produced during the glycolysis, and 2 NADH2 molecules also. Then next step is Krebs cycle. And before starting this Krebs cycle, there is one more interconnecting stage in which a decarboxylation reaction takes place, and that is a mediating step between the glycolysis and the Krebs cycle, or it is a connecting step between the glycolysis and Krebs cycle. Why means this pyruvic acid, which is the end product of a glycolysis, cannot enter into the Krebs cycle directly. Why? Because it is the acid which cannot take part in this Krebs cycle directly. So this pyruvic acid, by undergoing decarboxylation reaction, it becomes a 2-carbon compound which is added with the coenzyme A and becomes a acetyl coenzyme A, which is a 2C compound, and 1 carbon that will be released in the form of carbon dioxide. There one more Na, that means 2 NADH2 molecules are produced in this interconnecting reaction, that is decarboxylation. So what is the main intention of this interconnecting stage of between the glycolysis and Krebs cycle is pyruvic acid molecules. When two molecules are produced as an end product of glycolysis, so for each pyruvic acid molecule, if it enters in, that means if the pyruvic acid continues its changes through Krebs cycle, then definitely that should undergo this carboxylation reaction, and this acid that should be changed into 2C compound acetyl coenzyme A, and at the time of this formation of acetyl coenzyme A, one CO2 molecule will be released in the form of CO2, and for one pyruvic acid, one NADH2 molecule will be produced. So there are 2 pyruvic acid molecules, and those 2 pyruvic acid molecules, they releases 2 NADH2 molecules. Okay, then next Krebs cycle. During the Krebs cycle, it produces number of energy currency molecules, and those are 2 GDP or GTP. GDP is converted into GTP. Then 6 NADH2 molecules and 2 FADH2 molecules. And we know that for one pyruvic acid molecule, 3 NADH2 and 1 FADH2 molecule, 1 GTP is produced. And there are 2 pyruvic acid molecules. That's why multiply this number into 2. Then you will get this number of currencies. So, what is why we are multiplying it with multiplying it with 2 means there are 2 pyruvic acid molecules and these 2 pyruvic acid for 1 pyruvic acid molecule Krebs cycle should turn into one time, and 2 pyruvic acid molecules are there. That's why the Krebs cycle should turn 2 times. Then only one glucose molecule will be converted into energy currencies as CO2H2O plus energy currency totally. That's why it is aerobic respiration. So Krebs cycle will be moved 2 times because of the 2 pyruvic acid molecules which are produced by the glycolysis from 1 glucose molecule. Then during the electron transport, what happens? That means actually this NADH2, FADH2 are not directly utilized. But this NADH2 by undergoing electron transport, it synthesizes ATP molecules through chemosynthesis. Okay, so here if we calculate the total number of NADH2 molecules during the glycolysis, 2 NADH2 molecules are produced and 4 ATP molecules. Then during the Krebs cycle, 6 NADH2 molecules are produced and 2 FADH2 molecules are produced, and during the decarboxylation or interlinking or intermediate stage between the glycolysis and Krebs cycle, there also 2 NADH2 molecules are produced. So here each NADH2, one NADH2 molecule gives 3 ATP molecules. So here 6 NADH2 molecules are there, which are multiplied with the 3, then we'll get 18 ATP molecules during the Krebs cycle, and 2 FADH2 molecules. From each FADH2 or one FADH2 releases 2 ATP molecules. That's why 2 FADH2 produces 4 ATP molecules. Then 2 NADH2 through this decarboxylation gives 6 ATP molecules. That means multiply it with 3. So here total number of this ATP molecules, they are 36 plus 2 GTP, and that means total 38 molecules are produced from 1 glucose molecule. And a cytoplasmic enzyme nucleoside diphosphate kinase that converts GTP into ATP. So what we should understand is this GTP molecules furtherly they are changed into ATP molecules in the presence of this nucleoside diphosphate kinase enzyme. So total 38 ATP molecules are produced from 1 glucose molecule. And in prokaryotic cells, oxidation of 1 glucose molecule always gives 38 ATP molecules, and then NADH2 is not to enter mitochondria, which are absent there. That means in prokaryotes, mitochondria cell organelles are absent. That's why this cannot enter into the mitochondria. But in eukaryotes, this ATP molecules they enters into mitochondria, and there they are stored. That's why these mitochondrial organelles are treated as powerhouses of the cell. Then overall result of aerobic respiration. That means complete oxidation of 1 glucose molecule results in the following products. What are they? See here, release of 6 CO2 molecules and utilization of 6 O2 molecules and formation of 12 H2O molecules in 3 different steps. That means glycolysis, Krebs cycle, and the electron transport. Throughout these three steps, the resultants and products are 6 CO2 molecules are released and 6 O2 molecules are utilized and formation of 12 H2O molecules. Here we can recall RQ. Respiration quotient. What is respiration quotient? Ratio between released molecules by absorbed molecules. Here, released molecules, carbon dioxide molecules are 6 and absorbed molecules, oxygen molecules are 6. So, for carbohydrates, RQ is 1. 6 by 6 is equal to 1. So, it is proved here. The next 12 H2O molecules are released or synthesized. So, overall process of aerobic respiration may be shown by the following equation. That means by showing this number, we can draw the chemical equation of aerobic respiration. That is C6H12O6 plus 6O2 gives rise 6CO2, 12H2O and 686 kilocalories of energy, which was with the practical, it was proved. The next, pentose phosphate pathway. Pentose phosphate pathway is an alternate method of aerobic respiration. We know the aerobic respiration very clear, but there is a substitute pathway which is called as pentose phosphate pathway, which occurs in the cytoplasm of mature plant cells. And this pathway accounts for 60% total respiration in liver cells, not in all, especially in 60% in liver cells, it is of 60%. In this, for every 6 molecules of glucose, 1 molecule is completely oxidized into carbon dioxide and reduced coenzymes, while 5 remaining carbon molecules are utilized for the regeneration of oxaloacetic acid. So that's why it is called as pentose phosphate pathway. Pentose means 5. 5 molecules, they are utilized in the regeneration of oxaloacetic acid, which is essential for the regeneration of this Krebs cycle or a continuous rotation of a Krebs cycle. That means out of 6 carbon molecules in a glucose molecule, in one glucose molecule, 6 carbon molecules are there. Out of 6, 1 molecule is totally oxidized during the aerobic respiration and results in the formation of carbon dioxide molecule and reduces coenzymes. And the remaining 5 carbon molecules, why? Because glucose is a 6C carbon compound. Out of 6, 1 carbon is utilized for the oxidation of and result in the formation of carbon dioxide and coenzymes, while 5 more are utilized for the regeneration of this oxaloacetic acid. Okay, so that's why it is called as pentose phosphate pathway. The next is fermentation. Fermentation is similar to anaerobic respiration. It means both are similar. The difference is, there is a little difference. Difference is that the substrate breakdown in fermentation is extracellular, whereas anaerobic is intracellular. That means this anaerobic respiration takes place within the cell. That's why it is intracellular. And fermentation that takes outside of the cell. That's why extracellular. And the 2 most common types of fermentations. There are 2 important types of fermentations. Those are alcoholic fermentation and the other is lactic acid fermentation. And ethyl alcohol is the end product of alcoholic fermentation. In alcoholic fermentation, the final product is ethyl alcohol, and lactic acid is the end product of lactic acid fermentation. So that is the difference. And if we observe alcoholic fermentation, it is mostly observable in yeast, which is a unicellular organism. Saccharomyces is its botanical name. So in this Saccharomyces or yeast, we can observe the alcoholic fermentation very easily. Then yeast cells, they releases one enzyme in surrounding medium, and that enzyme break down the substrate and takes place outside of the cell. This change that takes place outside of the cell, that's why it is extracellular. And here also end product is ethyl alcohol plus carbon dioxide plus energy, but energy is very less. In aerobic respiration, that energy is 686 kilocalories, whereas in this alcoholic fermentation or in this anaerobic respiration, this released energy, it is very less. That is in the form of where it is shown in the form of the equation. C6H12O6 and the absence of oxygen gives ethyl alcohol plus carbon dioxide plus 2 ATP molecules. That is a alcoholic fermentation, and this changes or described by French chemist Gay-Lussac and Gay-Lussac in 1815. He discovered and described all these changes. And in lactic acid fermentation, lactic acid is the end product and carried out by lactic acid bacteria. And this bacteria can ferment the lactose sugar formed in milk. That means in milk, lactose sugar is there, that is fermented by the bacteria. And this enzyme involved in the fermentation is lactic dehydrogenase. So whatever lactose sugar present in milk is that undergoes fermentation in the presence of an enzyme, that is lactic dehydrogenase, and by undergoing this fermentation, it produces and releases CO2, H2, and this bacterium is released. Okay, this enzyme is released by the bacterium microbe, and that helps in fermenting the lactose sugar into lactic acid. Okay, then another type of fermentation called acetic acid fermentation. This is one more type of fermentation besides this alcoholic fermentation and lactose uh, fermentation, lactic acid fermentation. There is another type of fermentation, that is acetic acid fermentation, which requires oxygen and is carried out by bacteria like as acetobacteria aceti. It occurs in two steps. First, glucose is converted into alcohol anaerobically. That means in the absence of oxygen, it is incompletely oxidized. That means glucose is incompletely oxidized and results in the formation of alcohol. Then next, this alcohol is oxidized into acetic acid by using oxygen. Now, C6H12O6 gives rise to C2H5OH, that is ethyl alcohol plus carbon dioxide. And this is anaerobic process. And second is aerobic process, that is ethyl alcohol by combining with oxygen results in the form of acetic acid plus H2O. CH3COOH is the acetic acid and H2O, both are produced. And microbial fermentation. Microbial fermentation is very useful for mankind. Why? Because this process is used in various industries to produce antibiotics, vitamins, dairy products, etc. All these industries, these microbes are playing an important role. Alcohol for the industrial and medicinal uses. Alcohols are used in medicinal preparations and bakery products. In the preparation of bakery products also, these microbes are helpful. And lactic acid, butyric acid, and acetic acid. Uh, all these are useful in different industries. And fermentation is also used for tanning of leather and incuring for tea and coffee. Yes, tanning means giving color to the goods. That means in leather industry, especially in leather industry, this fermentation process is used to change the colors for leather goods and tea, coffee also. Then faster effect. On this respiration process, one opinion is delivered by Pasteur, and on behalf of his name, this effect is called as Pasteur effect. And Pasteur, scientist, he observed that the rate of fermentation and breakdown of sugars was higher under anaerobic conditions as compared to aerobic conditions. That means what he observed or what are the observations by Pasteur is that he observed the fermentation process in sugars, and what he found is this sugars they releases energy, and that energy is showing difference. In which process this energy is releasing more means that change was observed by Pasteur, and that energy is released very in high in anaerobic conditions or under anaerobic conditions. The sugars they undergoes fermentation process. That means the breakdown of sugars takes place and releases more or higher amounts of energy when compared with the aerobic conditions. Material respiratory substrate is same, that is nothing but sugars only, and these sugars under anaerobic conditions they are releasing high amount of energy than the aerobic conditions. So by this we can understand that microbes they plays an important role in releasing energy also. So that's why in different industries these microbes are using microbes are utilized in different industries. And the anaerobic breakdown of sugars into carbon dioxide, ethyl alcohol due to the presence of O2 under aerobic conditions were termed as Pasteur effect. So, what is Pasteur effect? Means anaerobic breakdown of sugars into carbon dioxide and ethyl alcohol. Here we have shown in the equation form. Under anaerobic conditions, this glucose that splits into ethyl alcohol and carbon dioxide. Now this ethyl alcohol that respires or that by adding with oxygen or under aerobic conditions that releases what the this acetic acid and H2O. So this all these changes are togetherly called as Pasteur effect. Why means Pasteur only detected this change and he only noticed this change and published this points. That's why on behalf of his name, this change is called as Pasteur effect. What is Pasteur effect? It is nothing but release of energy by using sugars as respiratory substrates. And under anaerobic condition, these sugars releases high amount of energy than the aerobic conditions. That means anaerobic conditions are playing an important role by utilizing the microbes. That's why they releases more energy than the aerobic situations. So with this is the Pasteur effect, and then with this respiration process. It was completed. And next, what we have to discuss is factors affecting respiration and um, yeah, amphibolic pathway. That means how respiration is, why respiration is called as amphibolic pathway, and what are the factors that affects the rate of respiration, under which conditions respiration rate is changed, and what are the external factors and what are the internal factors. All those things are discussed in factors affecting respiration, and why the respiration is called as amphibolic pathway. That will be also that we will actually discussed in that next class. And so here today, what we have learned is summary of production of ATP molecules during the aerobic respiration. So total number of ATP molecules are 38, and how they are and where they are produced is discussed under this. Under glycolysis, it produces two NADH molecules and 4 ATP molecules, and in the intermediate or linking process, in the linking process of pyruvic acid, it releases two NADH2 molecules, and during the Krebs cycle, it releases two GTP and 6 NADH2 and 2 FADH2 molecules. And for each NADH2 molecule, it releases or it synthesizes 3 ATP molecules. That's why Krebs cycle 6 NADH2 are multiplied with 3, which gives 18 ATP molecules, and 1 FADH2 that releases 2 ATP molecules. So here 2 FADH2 molecules are there. So they are multiplied with 2, then 4 ATP molecules. These are 18 and these are 4, 22. And 2 GTP under the influence of diphosphate kinase enzyme, this GTP is changed into ATP. So total GTPs are 2. That means 2 ATP molecules are produced. Here 18 plus 4, 22 plus 2, 24. Then 2 NADH2 molecules from this 2 NADH2 from decarbosylation. So here 6 ATP molecules, and through TCA 2 FADH2, 4 ATP, and 6 NADH2 Krebs cycle. So in this way, total 36 ATP molecules are produced during the aerobic respiration. And in prokaryotes, especially, oxidation of glucose molecules gives 38 only, but they are not diffused into the mitochondria. Why? Because those organelles are absent in prokaryotes. Whereas in eukaryotes, this 38 ATP molecules are stored in the mitochondria. That's why this mitochondrial cell organelles are treated as powerhouse of the cell. Okay. Then next is overall result of aerobic respiration. As per the equation, C6H12O6 with reacting with oxygen results in the formation of carbon dioxide and H2O and 686 kilocalories of energy. And if we balance the equation, then we'll get the molecules which are released and which are utilized. So here, after balancing this equation, we can observe that the release of 6 CO2 molecules in the equation, and the utilization of 6 oxygen molecules in this, and the process and formation of 12 H2O molecules also be observed by this equation. Okay, so here released energy is 686 kilocalories. Then why it is called as pentose phosphate pathway? Means only one carbon molecule is utilized in the synthesis of for the oxidation of a carbon dioxide molecule, and 5 more molecules are utilized for the regeneration of the material or product. which helps in running the process again and again. That's why it is called as pentose pathway. And we know the fermentation, which is similar to the anaerobic respiration. And here, this difference between these two, fermentation and anaerobic respiration, is only one. That is, fermentation is an extracellular activity, whereas anaerobic respiration is intracellular activity. And in this fermentation, we have two types. One is alcoholic fermentation, and the other one is lactic acid fermentation. In alcoholic fermentation, these changes are observed by Gillisack in 1815, and the main important change is a glucose molecule under anaerobic conditions splits into or changes into ethyl alcohol plus carbon dioxide plus 2 ATP molecules plus 58 kilocalories of energy. When compared with the aerobic respiration, it is very less. Okay, then next, another type of fermentation is acetic acid fermentation. And this acetic acid fermentation, and this two steps are there. First, glucose undergoes anaerobic respiration to release what ethyl alcohol and carbon dioxide. Then, after this synthesis of ethyl alcohol, it respires aerobically. Then, by undergoing this aerobic respiration, it releases acetic acid with the water molecule. So here, what is the important point is microbial participation is utmost important thing, and which is useful for mankind. In different industries, these microbes are used. Okay, like medicinal, like bakery, like tanning industries, these microbes are very much helpful. And what is the Pasteur effect? Pasteur discovered and stated one thing. What is that? When this microbes first by undergoing anaerobic respiration, the sugars they splits into carbon dioxide and ethyl alcohol, and after that they undergoes aerobic respiration, isn't it? That means ethyl alcohol and CO2 are produced due to the presence of oxygen under aerobic conditions. That's why it is termed as Pasteur effect. So with this, the respiration, aerobic type of respiration is over, and tomorrow we'll discuss another topic. Till then, have a good day.